Good morning, everyone. And I'm really sorry that it was not technically possible to be with you physically for this conference. And I'm really sad about that because, you know, we started the QA Quantum Africa conference series in South Africa several years ago. And it's really nice to see that that now it's taken up an independent life and keeps going and getting stronger and stronger with every edition. Yeah, it was just a little bit unlucky that that COVID prevented us to stick to the original rhythm of the conference. But I'm sure that now that things are looking a little bit brighter on the COVID side, we will hopefully be able to have many more people joining in person at the next edition. So speaking of COVID, you see my talk has COVID in the title, yeah, and it just you know, I will share some crazy ideas that we played with, yeah, and you know, it's a little bit of a speculative talk, and at the moment we don't have any particular hard evidence that what we are proposing is right, yeah. But I will try to embed it in a little overview of quantum effects in biological systems and why maybe there is really an opportunity of doing something interesting as quantum physicists. Yeah? So this is more or less the agenda of my talk. And sorry, I have a little bit of problem with my voice today. I got a little bit of a cold a few days ago. I will start by briefly giving you a brief overview of the interest of physicists in quantum effects in biological systems, yeah, and show you that it goes back many years. Yeah. Then I will give a little overview of some mainstream quantum effects in biological systems, just to give you a taste of what is out there and what could be interesting, yeah, or is interesting. And then in the last third of the talk, I will show you a quantum mechanism that could be used to explain the infectiousness of the coronavirus, and potentially leading to some therapies to cure the problem. Yeah, but you know, this is very speculative, and you know, don't expect to go to the pharmacy tomorrow and get something along this line. Yeah, it might be completely wrong. Yeah, but it was fun nevertheless to do it. Okay, just to thank some of the people involved. Most of the work that I will present today has been initiated by Adriana Mare, who is the second lady from the left on your screen, yeah, and then Maria Schult did also some interesting work, and the work specifically that I will present today is mainly due to Bethany Adams, yeah, who is the lady on the right of the screen. Yeah, so this was at a photo that we took at that's why I show it at a quantum biology conference in Vienna several years ago. Okay, let me start with a brief history of quantum biology, yeah, or of quantum effects that have some relevance for biological systems. Yeah. So, and you know, I don't need to tell you, you know, quantum mechanics has been around for more than 100 years now, yeah, and since the 30s, yeah, people have started to branch into various after let's say the foundations were set by the founding fathers of quantum mechanics, yeah, starting with Max Planck and Erwin Schrödinger and all the others. Yeah. So, but already early in the 30s, people starting applying quantum mechanics to various branches of physics and non-physics. Yeah. And already in 1933, yeah, Niels Bohr, who featured prominently, yeah, in the history of quantum mechanics, yeah, he already gave a lecture that was then summarized in a brief article in Nature in 1933. So in 1st of April, so it could be an April's Fool's joke, yeah, I'm not sure. but I think that if you read the article, it sounds solid. Yeah. And he proposes that you see in the yellow highlighted text at the beginning of the article, that that the new theory might have created also new background for the discussion of the relation of physics to the problems of biology. Yeah? So already almost 100 years ago, people had already this idea that that quantum physics might play a role in biological system. Yeah. And one of the persons that also pushed along these lines was Pascual Jordan. Yeah. And he also wrote very early, the early 30s, articles, and even published a book where the word quantum biology appears for the first time. Yeah, so that's really very interesting. Yeah. So here is the German edition of the book of Pascual Jordan. Yeah, also in the early 30s. Sorry, 1943, I think, is the date of the translation to English. And somehow I couldn't find the proper bibliographical notice of the book. I just told the picture somewhere online. Yeah. So, and this is the book where for the first time Quantenbiologie, yeah, in German appears. So this is essentially the birthday of quantum biology. Yeah? And it's really very interesting that so long ago people started thinking about things. Yeah? But the real push for quantum physics and biology came probably with the book by Erwin Schrödinger, yeah? Who also gave a series of lectures in the early 40s when he was in Dublin, and they were published as a book, and it's certainly still worthwhile reading, yeah, because it's really very interesting, yeah. And some of the things that that Erwin Schrödinger sort of foresaw, yeah, was that the quantum nature of the energy levels might have a substantial role in understanding the genetic structure and the stability of our genes and so on and so forth. Yeah? And in fact, he envisioned that our what we know now is our DNA, yeah, in helix form was some kind of quasi-crystal or something like that. So he had a kind of a intuitive picture of what would be discovered properly, probably some 10, 20 years, 10, 15 years later by Watson and Crick and so on and so forth. Yeah. So what he did not consider, yeah, was in his book was the more interesting properties of quantum mechanics that we are also fond about these days, yeah, where we speak about quantum computers and quantum cryptography, quantum metrology, and blah blah blah. Yeah, so he didn't really think that tunneling. So in the book there is no really reference to tunneling or to quantum coherence or to entanglement. Yeah? Although Schrödinger was the one who understood that entanglement is a crucial characteristic of quantum physics. Yeah? But nevertheless, it was really a very interesting look into the peek into the future. Yeah? So now, these days, what do we understand under quantum biology? Yeah? Probably, you know, quantum biology sounds fancy. Yeah? It would be probably much better always to speak of quantum effects in biological systems. Yeah? And very funnily, if you look up on Wikipedia, the definition is taken out of a review article that we wrote some years ago. Is inspired by that. Yeah. So this is more or less what we understand under quantum effects and under biological into biological systems. Yeah. And you know, there is also this more humoristic approach to explain the necessity of quantum biology. Yeah, life is a molecular processes are quantum, therefore life is quantum. Yeah, so whether you believe in this kind of logic or not, I leave it to you. Yeah, but you know, there are serious and less serious approaches to try to define what quantum biology is about. Yeah. So what I want to do now, sorry, do I need to check? Sorry, if there is something in the chat, but somehow I can't reach it. Anyway, if there is something urgent in the chat, just let me know. Yeah, somehow my cursor disappeared now. Anyway, you see the history of applying quantum mechanics to chemical and biological systems started essentially with the discovery of quantum mechanics. There, people started thinking about using quantum theory, people like Heitler and London to describe the theory of chemical bonding. Yeah, then there's a very famous work by Born and Oppenheimer on molecular wave functions. Yeah, then already still in the 20s. Yeah. Hund and Mulliken started developing the molecular orbital theory. So you see, we're slowly the theory quickly built up, yeah, to describing the complex molecules, yeah? Alpha tunneling and so on and so forth, yeah. And then in the 30s, it started getting a little bit more serious, yeah, because the Journal of Chemical Physics was founded. Yeah? So as a kind of a recognition that physics can contribute a lot to chemistry at the foundational level. Then hydrogen tunneling was observed, and then at the same time there was the big discussion on quantum correlations, entanglement, non-locality, and so on and so forth. Yeah. And in the 30s, Max Delbrück started looking into mutations and genetic structures. Yeah? And the very interesting thing is that this happened after he read the papers by Bohr and Pascual Jordan that I mentioned before. Yeah, so he was in those talks, yeah, and thought, okay, this sounds really interesting. Let's get out of physics and into chemistry and biology. Yeah, so that's what he did, and I think it was a good move for his career. Yeah. And these early developments culminated with the book by Erwin Schrödinger, What is Life, in the 40s. Yeah. So these were kind of the kickstarting of the theory, the preparation of all the tools that we are still using today, most of them. Yeah. And then in the 60s, the sort of experimental evidence started to flow in. Yeah? In these very famous works in the early 60s, photoisomerization became known. Yeah, electron tunneling in photosynthesis was studied with using first laser experiments. Yeah, the hydrogen tunneling in enzyme reaction was also observed. And then we jump in the early 2000s with the really landmark work by Fleming and Engel and many others in subsequent years, where for the first time quantum coherence in the energy electron transport in photosynthesis or in light harvesting complexes was observed for the first time. Now, not everybody believed that it was correct at the time, but many more experiments have been done in that since then. Yeah, even experiments that showed that this quantum coherence survives up to room temperature. Yeah, so which is really counterintuitive, because we all assume that quantum mechanics essentially works very well at low temperatures. At the moment you reach wet and hot systems, the coherence should have kicked in. Yeah, but there is some evidence that is not always the case. Yeah. Okay, so this is the really an outlet. And since the 2010, activities exploded, and I didn't even try to summarize them explicitly. Yeah? From the theory side, some of the highlights, for instance, the famous work by Roger Penrose and Stuart Hameroff, yeah, who suggested, sorry, and the photo covers some of the captions, yeah, that quantum coherence might play a role in how our brain works. Yeah? So this is, of course, also quite speculative, and there is not real experimental evidence so far, as far as I know. But these are nevertheless very interesting ideas that kickstarted the whole phenomenon of looking for quantum effects in the brain. Yeah? Relevant for us, for the discussion that will come later, is probably the pioneering work of Luca Turin, yeah, who proposed an alternative mechanism to olfaction. Yeah, you know, the standard approach to explaining how our noses work, yeah, is a kind of lock and key mechanism, yeah, where the substance that you want to smell docks to the receptor that tells you what you smell, yeah. And Luca Turing proposed a theory where instead of where this lock and key mechanism is replaced by a tunneling phenomenon, yeah. And the idea is very appealing to me because, you know, if you think that, you know, we're all the product of evolution, it will be difficult to explain how we can smell something that hasn't been around for thousands, if not millions of years, yeah, because we wouldn't have the receptors in our brain. Yeah? So if someone develops a new perfume that nobody has smelled before, yeah, it's not very likely that we have that receptor. Yeah, so it's very interesting that you can in principle replace the standard approach, the standard modeling, basing it on a tunneling approach that is way more flexible. Yeah, because it's only a matter of frequencies. Yeah, if you want. So this we will discuss in a little bit more detail later, and I need to check the time because I'm speaking too much, I think. Another very relevant work, which is relevant for our discussion as well, is the proposed mechanism based on radical pairs to explain how birds navigate, yeah, when they migrate from the northern hemisphere to the southern hemisphere, yeah, and that is also very very interesting potential quantum effect, yeah. And then of course other coherent transport effects in ion channels and so on and so forth, yeah. And after 2010, many more approaches and theories and blah 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 have been developed, yeah. But I don't want to confuse you with too much, and probably confuse you too much anyway, yeah. Anyway, so that was a sort of historical embedding to show you that interest in quantum effects in biological systems is nothing new. Yeah? It has been around for a long time, almost as old as quantum mechanics. So let me give you a very brief overview of quantum effects in biological systems. Yeah? If we try to classify it a little bit, yeah? And this is reproduced after a table in one of the early reviews 10 years ago by Fleming, Scholes, and others. Yeah? You see, we can look at classifying the biological systems, the biological phenomena, and the quantum processes. Yeah. So, and essentially, we have three physical setups: radical pairs, like matter interaction, if you want, and various excited excitonic electron transfer states, and so on and so forth. Yeah. So this is some of the phenomena that play a role. Yeah. Essentially tunneling, production of radical pairs, and electron transfer, and so on and so forth. Yeah. What is also interesting is to look at the time scales that are involved. Yeah, because some of these phenomena might be a little bit counterintuitive. Yeah. So if you think of, for instance, the coherent transfer of energy or in photosynthetic setups, yeah, we speak of processes at the femtopicosecond scale. Yeah. The radical pair mechanism, yeah, is on the scale of microseconds. Yeah. If you look at the potential quantum effects that might explain how our neurons fire, yeah, or the catalysis of enzyme and things like that, we are speaking of milliseconds. Yeah. But what people really would like is to go to seconds and even hours. Yeah. And there are some proposed mechanisms. Yeah. Like for instance, there are molecules called Posner molecules that contain phosphorus in a kind of in a cage that protects its coherence. Yeah, and possibly we might have them in our brain. Yeah, and those effects have a chance of lasting maybe not hours, but several seconds. But these are things that are also a little bit speculative, and several people are trying very hard to find the experimental evidence that this is indeed possible. Also interesting, it is also an old slide, to living in a quantum world. Is about a Scientific American article by Vlatko Vedral, in which he listed that over the years we started measuring entanglement in warm and wet systems, which it's also, as I said before, a little bit counterintuitive. So let me give you briefly a little bit more detail about some of these quantum effects, and then I will speak about our model for COVID. So the model of photosynthesis is very simple. You have a photon hits an antenna, then the excitation jumps a little bit around until it reaches the reaction center, where then electrons are transferred, and so on and so forth. Let me skip a little bit. Because I see the time is already very advanced. This is just the structure of these photosynthetic complexes, and this FMO, Fender-Matthew-Olson complex, is where all the action takes place. And these are some of the first results of the paper by Fleming and others that I mentioned at the very beginning, where there is some evidence, claimed evidence, that coherence effect play a role in the energy transfer of the excitations. So this is very was very interesting and very surprising. And people then started investigating the pathways of the energy transfer. And so on and so forth. And so, and I don't remember, so I was looking, you see, let me jump to here. And then people got a little bit too excited, because they said, okay, now we have green quantum computers. So that maybe was shooting a little bit from the hip. But anyway, that just to explain to you that the buzz that was experienced when these experiments were done for the first time. And then, of course, people started developing models for these things, and provide the theoretical background for hoping to find entanglement in these systems. Okay, let's skip. The other big class of quantum effects in biological system is magnetoreception. So people know that little birds like the European robin can migrate over thousands, if not 10,000s of kilometers. And if you look at some of the graphs that you can find easily on the internet, or in publication, they travel along straight lines. So they know exactly where to go, and they get there. So that's really very interesting. And people started looking for mechanism to explain how this works. And this is just a figure out of a publication by the Vilchko family, that had been studying this phenomenon for many years. And you see that in the graph A, you see the orientation of the geomagnetic field and how the bird orients itself. So the experiments are really done putting a bird in a kind of cage and seeing if the bird moves towards follows the magnetic field lines. And you see if artificially the magnetic field is inverted, the bird goes in the other direction. And then you see also the interesting, if you disturb the signal with some radio frequency, then the bird is lost and doesn't know anymore where to go. It becomes a random process. And interesting also to understand the mechanism. If you anesthetize the beak of the bird, it doesn't really change the effect. So it's an effect that is independent of the state of the bird, and so on and so forth. There are many more cases. But so this is quite the primary evidence that birds follow magnetic fields. And the mechanism is fairly straightforward. Yeah? The bird follows the magnetic, so essentially it moves orthogonal to the magnetic field lines. A photon hits sorry, it took me a while to understand what CRY means, but ER stands for European Robin. Yeah, so there is a system in all these things where then the radical pair is formed, and depending whether it is in a singlet or in a triplet states, a process get kick-started, and the bird knows whether he needs to follow that line or he needs to correct its trajectory. Yeah. So this is really very interesting. Yeah. And what is also interesting is that you really need a photon. Yeah. Without the photon, it wouldn't work. Yeah. So here's another maybe more schematic version of the process. There you have again your photon. It hits the donor molecule. It creates the electron pair, and so on and so forth. And then quantum mechanics, hyperfan interactions kick in, and you end up having these singlet or triplet states that that then allow the bird to understand what it has to do. Yeah. And then okay, here more experimental and evidence on the sensitivity on the magnetic field and so on and so forth. Yeah. Okay, and this is also out of a fairly recent publication where people start believing that the mechanism that birds use could be used also in technical applications, technological applications, to develop very sensitive magnetic devices. Yeah. Okay. Now, since it's already 11, let me come quickly to the real topic of the talk. Yeah. And what I would like to show you that to see whether quantum tunneling might play a role in this system. And this is work done with Brittany Adams that I mentioned, Ilya Sinaisky, who worked many years with me at QA6-Third in Durban, and Rin van Gondelle, who worked also for many years in quantum biology and who made sure that our biology was right. Yeah. Yeah, we were not worried that our Hamiltonians are right, but we were worried that our biology would be completely wrong. Yeah. So again, sorry, maybe I repeat myself a little bit with a different touch on slides that I showed before. But what we want to do now is, if you look at the bottom line of this table, we want to see whether we can do a theory that is similar to the olfaction theory proposed by Luca Turin several years ago, which is based on a kind of a vibration-assisted or induced tunneling, and applied to the current pandemic in a way or another. Yeah? So, and the idea is very simple. Yeah, several years ago, we already started looking at the effects on vibration on energy transfer. Yeah, and the simplest energy transfer system is this kind of dimer system. Yeah, and if you do the maths, yeah, you can see that you can find an easy expression for to calculate the probability for the transition from state 1 to state 2. Yeah, that's very straightforward. Yeah, and this you can generalize a little bit more to a dimer coupled to two different environments. Yeah, and if you do the whole maths within this canonical theory of open quantum systems, so you see you have the same dimer system that we had in the previous slide, except that now the state 1 and the state 2 are coupled to two spin bars. Yeah, that somehow model roughly the setup of magnetoreception, if you want, yeah, for the quantum tunneling that we want to discuss now. So yeah, and then you can, I mean, the model becomes a little bit more involved, so that you need to solve it now numerically. And what you can see is that there are, if you do a plot where you listen couple the decay rates and the and the time for the transition, you quickly discover that there are regions where coupling the dimer to two baths increases the probability for the transition for certain parameter regimes. Yeah? And you can do it even for correlated baths, which is probably even more realistic. Yeah? And the regions becomes even bigger. Yeah? So there is really a potential mechanism there to do kind of a tunneling assistant transport, exploiting the interaction of your dimer with the surroundings. Yeah, so that's so. And we tried, and then we applied this approach to the famous FMO system, really making an effort to use really biological relevant parameters, yeah, so that essentially getting the data from the experiment, and the effect is still there even in those parameter regimes. Yeah. Okay, so so now back to we need to take the bend because okay, to COVID. Yeah, let me skip this because I think I'm running a little bit late. Let me skip this. Okay, and let's look at this. Yeah, so this was in a cartoon, the basic idea of the vibration assistant tunneling process. Yeah, and one of the reasons why people are interested in this is that because we know, for instance, that if you change the isotopes of what you want to smell, yeah, you might not be able to smell them. Yeah, so there are really funny phenomena occurring. Yeah, and also people know that the same mechanism, yeah, probably applies also for neurotransmitters. Yeah, for instance, you can see here that serotonin and LSD, yeah, that have probably similar effects, share a similar tunneling spectra. Yeah, so that this is a kind of indirect evidence that there might be something there. Yeah, and what we are discussing now, it falls essentially in the same class of biological system. Yeah, because here we have also the SARS-CoV spike that wants to bind to your ACE2 and so on and so forth. Yeah. So the picture that we had in mind when we started this was essentially to replace the lock and key mechanism with a kind of a plug idea. Yeah. And this cartoon, maybe I can show it. Sorry, I can show it again. Yeah, it's what it sort of tries to indicate. Yeah, I don't know if it's a very nice description, but it's just that the vibrations induce the tunneling, and the two things come together more like a plug than a key and lock mechanism. Yeah. Okay, so and this is the very simple model. Yeah, and almost ashamed to share it. Yeah. The simple model is here. We have our spike protein described by a parameter omega, and we have our donor acceptor system. Yeah, so that the quantum model is really very simple. You see this again, it's a dimer model like the one that I showed you before. Yeah, exactly the same. Yeah, and we can do then also essentially exactly the same. Yeah, we couple this to an environment. Yeah, and we calculate again the probability for the transition for the plug to attach to the receptor. Yeah, yeah, and this is the paper is in the archive and it should be published soon. Yeah, yeah. So this is the Hamiltonian. Yeah, but. Okay, we have Hamiltonian, but we have zero. We had at that moment zero idea how to find any relevant parameters to try to make any prediction. Yeah, and so we, thanks to Bethany, who read a lot of papers. Yeah, she managed to find papers where the vibrational spectrum of the spike proteins was measured. Yeah. Through Raman spectroscopy or something like that. Yeah, And we sort of read this data and tried to extract the relevant frequencies to introduce in our simple model. Yeah, and we picked two vibronic modes that you can see here in the Raman spectrum in figure B. Yeah, around 1600 hertz. Yeah? And here you see what happens. Yeah, we just plot also a numerical study. Yeah, very parameters. Yeah, the tuning and the coupling strengths, and we looked for areas where this transition probability is maximal. Yeah, and you see that already in figure 1, there is an area, the blue area, where the probability for the transition increases. Yeah? And for slightly different parameters, yeah, the region becomes even bigger. Yeah, and if you play again, then you risk losing the whole effect. Yeah, for other vibration modes, these were just kind of experimental computational physics. Yeah, for the 100 inverse centimeter vibronic mode, it's even more interesting because it seems that there are two regions where the probability increases significantly. Yeah, and then of course after a while it disappears as well, diffuses away if you tune the parameters of this sweet spot. Yeah, so that in principle it looks like, yeah, that in this very simple model that the vibrations of the spike protein from the COVID virus, yeah, might enhance the electron transport. Yeah, and if it now really becomes speculative, yeah, you can turn it around and say maybe you can use vibrations to counteract the effect of the infection with COVID. Yeah, and there is some evidence that that this kind of electron transfer might happen in the COVID receptor, yeah, because there are similar classes of proteins where some evidence has been found, yeah. And the other interesting aspect is that people know that there are certain animals, yeah, that are resistant to COVID. They just don't get infected. And there are also humans that apparently still haven't been infected by COVID. Yeah? And that seems to have something to do with some redox activity and probably some lack of certain vibrations in certain hosts, yeah, if you want so, yeah. So, but this is also, you know, just in very vague indirect evidence. Yeah? So, but the implications are probably significant, yeah, because first of all, it could be a very interesting new mechanism to explore, yeah. Secondly, you know, you could come up with alternative treatments, yeah, maybe by using maybe light at the right frequency to counteract the effect of the COVID virus invasion, yeah, and maybe even to come up with new pharmaceutical on the long term, yeah. Now, what we have in mind is that, you know, you could just by Raman spectroscopy find out whether a new mutation of the COVID vaccine, of the COVID virus is more infectious than another one. Yeah, things like this that could probably very cheaply be implemented. Yeah, but you know, as I said, I cannot prove this, and maybe I might never be able to. Yeah, but it's nevertheless very interesting to think about these things. Yeah. So thank you very much. Sorry, I also went a few minutes overboard, and I hope I didn't steal too much time of your well-deserved coffee break. Thank you.
Well, thanks for the talk. Thanks. Any questions there from the audience in Kigali? Let me see. There is there are some questions here. Christian Aime Nejumen Thank you for this great talk. Which mechanisms of fighting SARS-CoV-2 are implemented inside vaccines nowadays, and what are their impacts on the problem of binding spike protein as you presented us? Well, that seems like a bit technical.
Certainly not this vibration system tunneling mechanism. Yeah, a more traditional lock and key mechanism. Yeah, if you were to do the really microscopic simulations. Yeah, and that is something that we want to do now. We want really to do proper density functional theory simulation to see whether there is any chance that the mechanism that we propose is there. Yeah, we just created a simple model and fitted some parameters. Yeah, but to still work on this, we need to come up with more solid arguments. Yeah.
Another question by Maura Telmini. Thank you, Francesco, for this wonderful overview. I have a question: how to bridge the gap between the microscopic phenomena at the molecular level, which are well handled by quantum mechanics, and the global body scale behavior of living entities, including emotions and consciousness? Okay, this is getting difficult here. How coherence might be preserved? So that's, and then, essentially, this is like a, okay.
Okay, that's the same copy and paste.
Exactly, copy. So, yeah. But I think
this is probably for another a question for another talk. But the short answer is that it seems like nature came up with mechanism to protect, let's say, an entanglement even in wet and hot environments. Yeah. And what is really very interesting, and I mentioned it in passing, is that there are these molecules called Posner molecules, yeah, that seems to have exactly this property, that this sort of the cage around a central phosphoatom, yeah, sort of protects the coherence fairly well, even in hot and wet environments. Yeah. But there is not yet much experimental evidence that that our brain works according to that. Yeah, there might be some tunneling, something here, something there in quantum in our brain. Yeah? But whether consciousness is a consequence of quantum coherence, it would be lovely. Yeah, but I don't know.
But this is getting a bit on the speculative side. There is another talk on the chat. Talking about another very speculative paper by Roger Penrose. In his paper on consciousness mentioned the quantum state collapse in microtubules, which was, of course, was kind of controversial. Yeah. So what do you say about this, question mark? But,
yeah, I can say that I don't know. It's there's all very nice ideas, but as far as I know, there is no hardcore experimental evidence that it is like that. There are many experimental groups that try to work towards finding some evidence, yeah, but it will probably take still a long while to come with a quantum theory of consciousness, yeah, and maybe it will never work. I don't know. Yeah.
This is a problem.
That's something that I would discourage young people to work on. It's something that you can do when you see your retirement at the horizon, yeah, and so it's better to stick to Hamiltonians, open quantum systems, and do some shut up and calculate approaches. Yeah. And when you retire, you can start thinking about consciousness and things like that. Otherwise, it can easily become a career killer. Yeah.
Yeah, I agree with Francesco that for a young person to get into this field, there are too many speculations. When people retire, like Penrose must be in his 80s now, lately. So once you're old and then retired, and then you can just sit down and speculate. But for the younger people, I think the more focused efforts would be better. So, okay.
I said I was going to ask a question about consciousness quantum entanglement, but I stop. I don't ask now because I listen to your advice. Thank you very much.
The only hard fact that we know about quantum consciousness and quantum mechanics is that you can use noble gases to anesthetize people. But if you use so we know how to switch off consciousness, with certain gases. But if you use isotopes of those gases, it might not work. So so we know indirectly, that there might be some subtle quantum effects involving nuclear spins, that might induce or not induce the switching off of consciousness, like in anesthesia, that we know, that experiments have been done, and that is a fact, but that is how you switch it off, not how you switch it on, So, and nobody really knows exactly how it works, but there are some indirect hints that some quantum effect might play a role, in all of this, but to put the finger exactly on what, I think it's early days.
Okay, thank you very much again, Francesco. So let's thank Francesco for the talk.
Thank you.